In the 50s, Paul Zamecnik and his group didn't know about Crick's central dogma or his adaptor hypothesis. They were studying protein synthesis from a biochemical point of view. Zamecnik made an extract using rat liver cells. This extract was basically a water-based solution that contained all the parts from a culture of cells. Everything needed to make proteins was in this extract. To follow protein synthesis, radiolabeled amino acids was added to the cell-free extract. After incubating the reaction at body temperature, the tube was centrifuged. Newly made proteins, being heavier, would pellet at the bottom of the tube. Unincorporated amino acids, protein building blocks, being lighter, would remain in the supernatant. Sure enough, the radiolabel was present in the pellet, showing that new protein had been synthesized. Mingled with the labeled proteins in the pellet were large cellular structures. These were later named ribosomes. Ribosomes are made of both RNA and proteins and are the sites at which protein assembly occurs. While the RNA part of the ribosome, rRNA, is involved in protein synthesis, its role was unclear. However, Paul Zamecnik and Mahlon Hoagland identified another RNA molecule associated with the unincorporated amino acids in the supernatant. Amino acids are first attached to a low molecular weight, soluble, RNA in this same soluble fraction, and these amino acids are subsequently transferred to the protein made in the ribosomes. Zamecnik and Hoagland named these intermediary carrier molecules soluble RNA. On a visit to their lab, James Watson recognized that soluble RNA met the requirements of Crick's adaptor hypothesis. Each of the soluble RNAs could pair to its partner amino acid and ferry the amino acid to the ribosome for protein synthesis. Soluble RNA was later renamed transfer RNA, or tRNA, to better reflect this role. Paul Zamecnik first developed the cell-free extract system, which Marshall Nierenberg adapted to decipher the genetic code. Paul Zamecnik and Mahlon Hoagland also isolated activated tRNA, the adaptor, that shuttled amino acids to ribosomes for incorporation into new protein chains. Mahlon Hoagland, working in Paul's lab in 1953, focusing on how amino acids were activated or energized prior to their incorporation into proteins. This is done by enzymes that he found, later known as aminoacyl tRNA synthetases, in the soluble fraction of a cell.